back to the brain. So you've done your visual test. You verified that they're genetically susceptible. They have abnormal proteomic markers. You're thinking this is mold, but you're not quite sure. Thank you. And we want another tool to further verify what we're looking at. And when you recognize all of the cognitive dysfunction that these patients have, we wanted some way to evaluate in a very objective way brain structures. Our feeling was if these people were inflamed enough, we would begin to see brain-related changes. And sure enough, we found one in a very unique brain MRI tool called NeuroQuant. This is a volumetric brain scan, and it basically uses a mathematical model to take the data from a normal, non-contrast brain MRI. It's not a fancy MRI, it's your standard MRI. And it takes all the data and it actually blows it up into a perfect sphere, and that way we can normalize internal structures. This was originally designed to offer early diagnosis for Alzheimer's and Parkinson's. So they would look at the substantia nigra and the basal ganglia to see whether or not there were some atrophic changes there. And we took this tool and said, well, let's just start imaging people's brains who have CIRS. Let's see what we find. We thought that there would be some sort of uniform architectural changes in the brain, that if the symptoms are the same and the blood labs are essentially the same, probably same changes in the brain, a fingerprint. What we found was something different. So this was cleared in 2007 by the FDA. It takes 10 minutes to run the data through the software program. And what you'll find is that there are many, many, many MRI centers around the country that have licensed the NeuroQuant software. So what you do is you go to the website for NeuroQuant called Cortex, and you can type in your zip code, and it will tell you your closest NeuroQuant MRI center. There was a question about, well, what codes do I use? You use your normal codes if you're going to order a brain MRI. You just also ask for NeuroQuant. So you send them to the NeuroQuant center and you write non-contrast brain MRI with NeuroQuant. And they'll not only produce a normal brain MRI report, but they'll also give you the NeuroQuant component too. Now, just like the HLA, you're going to have to do a little extra work when you get the report back. It's not going to tell you everything that you need to know in terms of the final assessment. But the NeuroQuant is really helpful in that regard. So what we see are changes in volume. Some areas of the brain swell and other areas shrink. And I talked about that yesterday. These correlate with genomic changes because of TGF-beta1 and C4a and a few other markers. And so what's happening is these inflammatory proteins are bridging the blood-brain barrier and getting into those key centers and degrading those areas of the brain. So this is an important point. In the neurology literature, there was a recognition that sometimes there are brain insults or injuries where the brain shrinks. And we know that there are two pathways or mechanisms that are potentially activated. One is called dendritic pruning. So dendrites, as you all know, should be beautifully branched and high volume so they can receive information from lots of local neuron input. But what happens is if you create activation of NMD receptors and microglial cells and localized inflammatory proteins, the dendrites start to retract and they start to pull in and they become a line, they lose their branching, and therefore there's some volume loss here. It turns out that if you remove the insult, the dendrites can regrow and rebud and bloom. In other circumstances, though, it's actual true volume loss and atrophy, so it's like scarring of the brain. We won't know if there's volume loss initially, whether or not it's pruning or atrophy, until you treat them. So protecting the brain along the way, I think, is really important to give them the best chance for regrowth and rebudding. So you want to restore normal brain tissue. And we've shown that you can do that in pre and post studies. And so here you have these microglial cells that get triggered under CIRS, and they start degrading the dendrites, and you get retraction. Atrophy is worse, of course. Can't tell on the NeuroQuant because it's structural. This is not a functional MRI. So we're not doing SPECT scans or those sorts of things. We actually are looking at volume loss or volume gain from normal. So here's your typical MRI slice. Here's where it's segmented. And then this is the first component of the NeuroQuant report. Of course, they look at the hippocampi. because that's involved in memory related to dementia. See how all these things connect to each other? And they also look at the lateral ventricles. This is what the radiologist is going to evaluate. So they're trained to look at certain components of the NeuroQuant report. What we did instead is we actually began to categorize from normal the left and the right hemispheres by percent change, by volume. And what we discovered is that there were patterns in the deviations from normal. And these patterns seemed to matter because they were consistent with exposures. What was surprising is that the pattern expression was unique for the original exposure, that in fact the changes when an individual has been in a moldy environment is different than when patients have Lyme disease. We did not expect that. And so NeuroQuant added validity to the diagnosis of CIRS, but it also adds some refinement to the diagnosis to say, you know what, your C4A is 20,000, and sure enough, you have brain-related changes consistent with mold. So they fail the visual test, their proteomics are abnormal, their brain scan shows mold, and now you're really cooking. You know what you're dealing with, okay? So you verify these patients. And I've seen patient after patient after patient who was told they have Lyme disease, and yet their C4A is 20,000 and their brain scan shows mold. Because remember, the symptoms are the same. The other way to get into that problem is maybe they were diagnosed with Lyme, it turned on CIRS, and now they're sensitive to mold. And mold is their big current issue. So even though they might have eradicated their Lyme, that their nanotrap test is negative, but now their C4A is 20,000 and their brain scan showing mold. That there can be a sequence of exposures, and it's the mold that's maintaining the illness complex. And unfortunately, they fall into the trap saying, well, I had this positive exposure to Lyme, my tests were, clearly abnormal, I got treated with antibiotics or herbs, and, and I'm still sick, so I know I have Lyme. It must be the biofilm, or it must be these pesky babesia, which are so quote-unquote hard to treat, or whatever it is, right? And it turns out it's not that at all. It's a different exposure altogether. So really understanding where these patients are at are key. And so we would evaluate the left and the right hemispheres, and we found that there were very specific changes in different areas. And we know that radiologists just can't eyeball the brain and decide which volumes are too large and too small, that it has to be mathematically precise in that regard. So this is what we found in mold. The forebrain parenchyma, the cortical gray matter, and the hippocampus were all increased. The caudate was decreased, and the pallidum was increased as well. For Lyme, normal cortical gray matter, hippocampus, and caudate, but a small forebrain, small putamen, and a large thalamus and large cerebellum. So how do you get to that conclusion? Well, here is your scoring system from 0 to 2, 2 being worse and 0 being best. And so when you look at the NeuroQuant report, you go to the left and the right, and you give a score, 0, 1 to 2. If the forebrain parenchyma in this patient was 33, they get a 2. And if the cortical gray matter was 17.2, they get a 2. You just add up all the scores on the left and the right. You give a column for each. And anything above 4, a total score of 4, so 5 or above, means they're positive for mold. Okay?
NeuroQuant does that, right?
No, you have to do this. So what you're going to do, so let's go through it. So this patient on the right was 34.3 for their forebrain parenchyma. Okay? They were 34.3 for the right side of their forebrain parenchyma. So we go to the scoring system, and we say, whoa, 34.3. That's greater than 32.3, right? So they get a 2. What if instead their forebrain parenchyma was 31? They get a 0. So no change from normal. And if they had a score of 32, they'd get a 1. Right? So you do that on the left side and you do it on the right side. If either side is 5 or above, they meet criteria. And we've published a number of studies on this for the NeuroQuant. And here's your scoring system for Lyme, 0, 1, or 2. But we've changed the thresholds because this is the pattern that we saw in the data. So no other combinations are like this in neurology. We've looked. This is not MS. Okay? You will see white matter flares, but they're generally non-specific. But now, with the role of NeuroQuant, we can characterize these architectural changes. It's never been reported anywhere else, and it's been verified. And it's even, we're talking about this at the break really quickly, NeuroQuant has also recently been held up in court, so even higher legal standard, because the data is so good.
Do you know if you can run the NeuroQuant analysis on a previous MRI, or does it have to be done at the time?
Good question. So the MRI tech knows that the scanner has to have very specific settings to be able to run the NeuroQuant report. Those settings are dependent on the type of scanner, whether it's Siemens or GE or whatever it's going to be. When you go to the Cortex website, this is the company that developed NeuroQuant, they will give you a list of the scanner and the settings. You may get a little lucky and say, boy, this brain MRI that was done 6 months ago actually was from a Siemens and it was T2 weighted with blah, 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 and actually we can run a NeuroQuant. So you can send the CD to the company and they'll run the NeuroQuant for $100 for you. Otherwise, you're out of luck. You got to start over. All right, so we've gone almost all the way to the end of verifying the diagnosis. Visual test, blood labs, brain scan, symptoms, and what about the music coming off the DNA, the genomic component? So we use a next generation sequencer. We know that genes respond to a variety of stimuli and toxins. And so we get this sort of coordinated immune response, stress response, and ultimately tissue clearance. And what we're really caring about is not the DNA that's there, but the RNA that's being produced. So that's the music. That's the actual activity of the DNA that's being expressed, influenced by epigenetics and also reflecting the original insult. So we looked at the mitochondria. There are 900 genes that are part of the normal mitochondria. And we found, interestingly, that if you look at the pattern description of these 900 genes, downregulation is normal, turns out, in regular sort of genetic activity. So that got coded as a blue of various shades. And if you had genes that were upregulated, that was abnormal. So there was overexpression, loss of regulation of gene response. So transcription factors that are not working properly. And so that was characterized by sort of an orange or a red. And you can see that there's a predominance of upregulation. As I mentioned yesterday, and this is our raw data, these are 50 subjects that we did pre and post. So we assessed their genomic activity. In our Lyme population, 500 extra genes out of the 900 were inappropriately turned on. And in our mold population, 700 extra genes were inappropriately turned on. This might explain the difference in the NeuroQuant. In addition to that, the way we structured our research study is, and this is an important clinical pearl, these patients had already gone through the entire treatment process, meaning they've normalized their visual test, their blood labs were also now in the normal range, but they still had some symptoms. And it's because of this. They had their ongoing genomic response. So we're now at the last step of therapy. We know that they've cleared their Lyme, they've cleaned up their home, their VCS is normal, their lipase is negative, so on and so forth. Now they're ready to be given the last step of therapy, the nasal spray called vasoactive intestinal peptide. Remember I mentioned it's genetically active in the way that it binds to cell membranes. And we said, come back to us when you feel better. And out of our cohort of 50 subjects, some of them came back in 2 months, and some of them came back in 6 months. So somewhere between at least 2 and 6 months, maybe longer, patients need the VIP spray to normalize the final consequences of this illness. And so how do we know that? So we actually ran their genomics again once they returned and they said they felt normal, finally and fully for the first time. So what did their genomics look like? It did this. We turned off all their genes. And what we find also as a result of VIP spray is that their NeuroQuants normalize. So their brain heals and their genes get better and they return back to a normal human being. This has implications for traumatic brain injury and concussion, chronic stress and PTSD. neurodegenerative disorders, even overlap with cardiometabolic illnesses that have an inflammatory component that's inducing gene activation, that we are seeing overlap with a variety of different chronic illnesses. And so we're sort of wiggling our way through to actually create corrective changes that are very deep.
What is this next generation genomic testing? What is that?
So the question is, well, what is this test? It's called a next generation sequencer. There's a few on the market. One is called Illumina. That's the one that we use.
Illumina?
Illumina. So if you have an extra $300,000, you can buy an Illumina. We were looking under the sofa for extra change. So what it does is it allows us to do full genomic profiling in patients, and we target the transcriptome, which is the RNA, and that is to say the music that's directly coming off the DNA. And so we then started testing it in our patients. Now, it's not cheap, so it costs about $1,500 to run a full genomic profile in a patient to get this data, and we did it twice in this patient population. So we wanted to make sure that we had full corrective changes because we knew that there were still some residual symptoms even when you got to essentially the last step of therapy. So note, this is pre-VIP, meaning last step, and then post-VIP.
Do you see Parkinson's responding like this?
We do.
Does VIP help with dysautonomia?
The dysautonomias typically go away with this treatment therapy. But again, there's a sequence of events that we have to go through.
I think insurance covers it if you have cancer diagnosis. I know in New York it's Guardian 360 and they cover it.
That'd be great. So there's a comment that if you have a cancer diagnosis, maybe it's covered. We've never done that before. We just, we run our sequencer, and I have a transcriptomist, a PhD geneticist, that conducts all of our research for us. So this is the typical treatment sequence. This is what has been studied in terms of 2 randomized controlled trials. We begin with removal of exposure. We then give a binder to clear out the inflammagens. Once that occurs and they pass their visual test, we eradicate their MARCONs with a nasal spray. We correct their gut, including food sensitivities. We then correct their hormones and androgens. We then deal with their ADH and osmolality. We then reduce their MMP9, correct their VEGF, correct their C3A, C4A, TGF-beta1, and then we give them VIP. So there's an order or sequence that we have you go through so that you can verify at every step that the patient is getting better. This is the strict protocol that's been studied. Not very integrative medicine friendly. So with that being said, it also doesn't include some other ideas that I think are important to fully get these people back to normal. So back to that gut immune brain idea. So we know that when people are under stress and they're inflamed, their guts are leaky and they have excess histamine release. So they're establishing this sort of chronic inflammatory process. So this is the HPA axis. This is the sympathetic chain with direct input into the gut lining, as well as production of cortisol, which begins to degrade the tight junctions as well, in addition to low MSH. And when cortisol levels go up, people become Th2 dominant, so they're actually more histaminic and their mast cells are leakier. And so they are also leaky because of that excess histamine production. Conversely, just as an idea, if their cortisol levels have flattened, they're now Th1 dominant and they're more likely to be autoimmune. But in the early stages of stress, you can degrade the gut lining and that will trigger an inflammatory process. Once inflammation is established in the gut, we also have triggering of indoleamines. These are small enzymes in the liver and the lining of the gut that once they become active because of exposures, they degrade tryptophan down the kynurenine pathway and they start making excess quinolinic acid. So when I test my patient's urine, we can see often high levels of quinolinates, which are also directly neurotoxic. So it's not just enough that they have this kind of inflammatory storm, we also see these changes because of the stress and also gut-related inflammation that makes it even worse.
How do you measure quinolinates?
In the urine.
By which lab?
Genova will do
Organic
organic
acid?
acids. Also, don't forget, as I mentioned, cortisol is on a teeter-totter with the immune system. So as cortisol levels climb, we see suppression of IL-6, TNF-alpha, CRP, right? It's our endogenous anti-inflammatory hormone. This is why prednisone was created. So again, in this stance, people are Th2 dominant as well, which means they're more atopic, they're more histaminic, and as a catabolic hormone, they're degrading the hippocampus, they're degrading peripheral tissue, and their immune system is now weakened as a result. As the brain changes and the body becomes more metabolically disturbed and peripheral cortisol receptors are altered, we see this happening, where the brain literally says enough is enough. And so cortisol levels start to fall. And if you look at the work of Bruce McEwen, who's an excellent physiological psychologist, he's articulated a lot of these changes in the stress response and how this progression occurs from high to low. But as this sequence of events happens and cortisol levels drop, what he's saying is that there's a reduced, what he terms allostatic load, that the body's saying, I can't take a high stress response anymore. You're causing too much damage. So it purposely turns down the stress response as a result to protect itself from the ongoing effects of high levels of stress. But the problem is that the immune system becomes more liberated and unregulated as a result. So when people have low cortisol levels, they're Th1 dominant, but they have higher levels of IL-6 and TNF-alpha and CRP, and they have a greater propensity for autoimmune illnesses. And so this is a much worse metabolic state for people to be in. It's not adrenal fatigue. Hypocortisolism is a disaster. It means they fried their brain and they've liberated their immune system. It's terrible. So, and you can imagine if the brain has already been sensitized to high cortisol, if you start giving cortisol back to try and make them feel better, which you might, you're also further creating changes in the brain. Oh, never thought of that before. The other way people get into this state is the body is so brilliant, it's so smart. When we're encountering an immune threat, and let's say it is a chronic infection, it's all over the literature, Epstein-Barr and tick-borne illnesses, all sorts of things. How does the body liberate cell energy into the immune response? It purposely turns down cortisol. And so you might very well be looking at an individual who absolutely has CIRS, and their cortisol levels are low because the body's trying to compensate for a chronic infection. And you could imagine the worst thing you could do is try and raise cortisol levels, because all you're going to do is suppress that immune response and allow the bug to grow and thrive, because you're degrading their terrain. The body's doing this on purpose. It's sort of just holding fast. Maybe it can't get rid of the bug, but at least it's trying to contain it. So in some sense, the worst thing you could do is actually raise cortisol levels. Right? Wait a minute. I thought low cortisol is terrible. Well, people feel terrible, and it leads to all sorts of bad consequences. But it might be the wrong thing to do in that moment, and to sort of clear out the terrain and improve their microbiome and clear out infections and let that immune system come back to normal. And you will see cortisol levels come up on its own. So when I see patients with low cortisol, I think, did they fry their brain or do they have bugs? Right? That's what I think. And what are the consequences? We know when people have flattened their cortisol curve and they've liberated their immune system, if they have a concurrent illness, whatever illness that is, they have far worse outcomes. It doesn't matter what the condition might be. If they have concurrent breast cancer, they have higher risk of metastases and mortality. If they have concurrent heart disease, they have higher risk of MI and dying from that MI. If they have diabetes, they're going to have more drug use and poor outcomes. Flattened cortisol curve means an upregulated immune system. High IL-6, awful. Chronically elevated TNF-alpha. Right? So these patients are a metabolic mess. Interestingly, the psychiatrists have figured this one out. So when you look at sort of the physiological psychologists and psychiatrists, where they look at the metabolic component of mood disorders, recently they have said, oh, well, you know what, interestingly, when patients have high cortisol levels, that's easier to treat. They're actually more sensitive to our standard antidepressants. They're actually now classified as treatment-sensitive patients. They've also figured out that when they're in a deeper metabolic pattern and they flatten their cortisol curve, they have hypocortisolemia associated with depression, they call it atypical depression or treatment-resistant depression. They finally got it, that the brain is connected to the rest of the body. and that in fact what's happening is this teeter-totter relationship between the immune system and the stress response is permanent. Another sliding edge, just like gut immune brain. So when stress is up, immune is down, Th2 dominant. When cortisol levels are low and the immune system is high, Th1 dominant, high IL-6, high TNF alpha. Much worse, much, much worse. And you have to ask yourself, how'd they get there? And how am I going to dig them out? In addition to that, patients can carry around toxins. So what happens when you have high levels of mercury or arsenic or lead or cadmium or PCBs or phthalates, whatever it's going to be, they bind to various tissues, right? So heavy metals in particular have what's called tropism. They have high binding affinity. So for example, lead loves calcium receptors, and mercury loves nervous system tissue, and cadmium loves kidneys, and the thyroid, for example, that heavy metals will go to different components of the body. And so what happens is once they bind in to those receptors, they deactivate or bump off the cofactor or mineral or nutrient that's supposed to be part of that enzyme system. So lead will displace calcium. There's a nice series of studies to show that people with high lead levels that are in the bone will have normalized DEXA scans, even though their bones might be fragile. So toxic metals make everything worse and have been linked, interestingly, to a lot of the same symptoms. And what happens is we just see a worsening of this complex, this gut immune brain complex. Digestive issues, immune disturbance, central and peripheral nervous system problems. So it's bloating and gas and impaired immunity and brain fog and depression and interrupted sleep and fatigue. So here is another way of organizing those labs. So diamine oxidase, which is an enzyme that breaks down histamine in the gut. Lipopolysaccharide. This is an inflammatory protein that binds to toll-like receptors that causes systemic inflammation, but it's generated by bacterial fragments in the gut. Melanocyte-stimulating hormone, because it helps to control tight junctions in the gut. You can do food sensitivity testing or GI effects tests. Here's all your immune testing. Is it general inflammation? Is it an autoimmune process like a positive ANA or rheumatoid factor? Do they have measurable toxins and viruses like heavy metals and chemicals or bugs? And then what about their innate immune response? MMP9, C3A, C4A, TGF-beta1, anticardiolipin antibodies, von Willebrand's, D-dimer. And then also, what about the brain? Well, abnormal cortisol curves for hippocampal atrophy or beta-2 microglobulin, which has been linked with blood-brain barrier deficits. Do they have a SNP for MTHFR? Plus minus, but okay. VIP, MSH, VEGF, TGF-beta1, C4A, all important for brain health and neuroinflammation. Have you done your NeuroQuant, your volumetric brain scan, and have they failed the visual test? So you have all of these tools now at your disposal to begin to categorize these patients and say, absolutely, you're a CIRS patient, and another lens, your gut immune brain. And we load on toxins and bugs on top of this inflammatory process, and it's even worse. So we also know that the presence of these chemicals in particular can shift cell metabolism to another form of abnormal cell response, which is cancer. So again, as the cell becomes more acidic, it is driven into the glycolytic pathway, making excess lactic acid, lifts it from the basement membrane, prevents the immune system from getting in and binding to it and killing it. So what do? This is just a list of all the other supplements that I might use for gut immune brain. So memorize this. And then what do I do about toxins? Well, I think about another triad, liver, lymph, kidney, which is our drainage organ. So for the homeopaths in the room, I really care about drainage. And so we care about the ability of the body to sort of have deep reserve and open enzyme systems that, the nutrients that you're giving them can sort of bind in and do their job. Some of this is driven by exposure. Some of it is driven by genetics. Interestingly, there are mechanisms by microbes themselves that actually tell the body to hold on to toxins. Candida is famous for this. There's a whole body of literature that if they have a candida problem, they're actually holding on to mercury. And they do this on purpose to help deactivate the immune response. So sometimes to get rid of a chronic infection, you do have to start detox in these patients. So these are some of the things that I do to help drain the body of toxins around liver, lymph, kidney. This is a bit beyond our discussion for today. So what are our treatment challenges in this patient population? Well, you have to deal with eliminating the exposure. So clean up the mold, kill the bugs, do whatever you need to do, clean up the dentition and make sure that it's gone. So your ERMI swab, your nanotrap, whatever it's going to be. You need to correct the inflammatory response in a sequence of steps. You have to repair the nervous system and restore normal genomic activity. So there's a whole series of things that we need to do in that regard. So we've gone through this process of how do we sort of vet patients to say, yep, you're a biotoxin illness sufferer, and now we need a plan to get them better. So the treatment steps, again, this is the sequence of events that is standard, and this is the sequence of events that we'll go through on how to correct all of these issues in order to make sure that when they end with VIP, everything has gone back to normal. Again, this is sort of integrative medicine 2.0, but what's amazing about the process is as the patient improves and they start finally feeling better, it's an amazing thing to behold. So there's an order to all of this. We start with foundational treatments to kind of just build their reserves and get them going. We then start dealing with exposure elimination, and that could be infections or mold. We then switch to reducing their toxin burden, and that means we want them to pass their visual test, clear their MARCONs, and replete their nutrient pools. We then work for their metabolic balance, which means beginning to detoxify them of heavy metals and chemicals, balance their hormones, balance their electrolytes, and then finally resolution and repair. So we stop degradation of their enzyme substrates, so basically MMP9. We restore normal innate immune response with C3A, C4A, and TGF-beta1. And then we work on their central nervous system and their genomics with VIP. All of this then becomes part and parcel to restoring normal physiology in this patient population.